Omega-6 and omega-3 essential fatty acids are both required in our diet. Our bodies do not make or formulate these essential fatty acids, and they must come from our diet. Omega-6s are oxidative, and omega-3s are antioxidative. Both of these play a crucial and beneficial role in cell function and energy production. However, a balanced ratio of 1 omega-6 to 1 omega-3 is required for omega-6s to maintain their beneficial role. When the oxidation load from omega-6s begins to exceed the antioxidation capacity of the omega-3s and our endogenous enzymes that help detoxify our cells, then cellular structure and functions break down. Cellular functions are diminished, our metabolic toxins begin to accumulate in our cells, and this leads to metabolic dysfunction and degenerative diseases. As an overview, here are a list of the omega-6 and omega-3 essential fatty acids. Oxidative are the omega-6 fatty acids, linoleic acid, or LA, which is an 18-carbon chain essential fatty acid. Antioxidative is omega-3, alpha linolenic acid, also an 18-carbon chain essential fatty acid. Both omega-6s and omega-3s are precursors for additional omega-6 and omega-3 fatty acids. On the omega-6 side, arachidonic acid is more commonly known. On the omega-3 side, you have EPA, which is eicosapentaenoic acid, which is what we get from fish, and docosahexaenoic acid, DHA, another well-known omega-3 antioxidant fatty acid. The key is controlled and balanced oxidization. There's a crucial role for omega-6s and cellular oxidation within our bodies and our immune system. Cellular respiration produces oxidization, which is necessary, and it is unavoidable. Cellular respiration is really no different than an internal engine's combustion. They both create heat and they both create exhaust. As a result of energy being produced in our cells and our mitochondria, oxidative components such as free radicals in the form of hydrogen peroxide and reactive oxygen species such as superoxide are also used by our body in very beneficial and essential ways. The immune system uses the cells destroying properties of free radicals to kill pathogens by engulfing an invader such as a bacteria or a virus, and then exposing it to a free radical such as hydrogen peroxide, which destroys the membrane of the bacteria or virus. Free radicals such as hydrogen peroxide act as a signaling molecule that calls immune cells to an injury site, aiding in tissue repair. Our thyroid gland synthesizes its own hydrogen peroxide, which is required for the production of thyroid hormones. Reactive oxygen species and reactive nitrogen species interact with proteins in cells to produce signaling molecules. These free radical nitric oxide have been found to help dilate blood vessels and act as a chemical messenger in the brain. By acting as signal molecules, free radicals are involved in the control of their own synthesis, stress response, regulation of cell growth, metabolism, and death. It also controls our cellular temperature, keeping our body at 98.6. Cellular oxidation regulates temperatures such as fevers in our immune response. Our body uses controlled oxidation in the process of apoptosis, the homeostasis of the cell, and also programs cell death, which is used to destroy cells that cannot be repaired or cells that begin to grow abnormally, such as cancerous cells. Just as our immune system uses the oxidative properties of reactive oxygen species and free radicals to destroy cells of pathogenic bacteria and viruses, and abnormal and damaged cells, excessive and uncontrolled reactive oxygen species and free radicals have the same power to destroy our healthy cell structure membranes and the internal components, which then damage cellular functions and signaling communications, and this will result in disease. Excess omega-6s in the Western diet induces nutrient deficiencies, and this causes a catastrophic lipid peroxidation cascade. This damages a phospholipid called cardiolipin inside the mitochondrial membranes. This leads to electron transport chain failure, and this causes mitochondrial failure and dysfunction. This leads first to reactive oxygen species, which feed back into this peroxidization cascade. So you're filling up your fat cells and your mitochondrial membranes with omega-6s, and these are going to pre-oxidize, because they are polyunsaturated. The next thing that happens is insulin resistance, which leads to metabolic syndrome, type 2 diabetes, non-alcoholic fatty liver disease. When the mitochondria fail, you get reduced fatty acid beta oxidization, meaning that you can't burn these fats properly for fuel. So now you're carb dependent and you're headed for obesity. So you're feeling tired, you're gaining weight, your mitochondria are failing to burn fat for fuel. This is a powerful mechanism for obesity. Again, Dr. Chris Kenobi and Dr. Mercola. The energy failure at the cellular level leads to nuclear mitochondrial DNA mutations, and this leads to cancer. 3 weeks on a high PUFA diet caused heart failure in rats. This also leads to apoptosis, or cell death, and necrosis, which is cell death due to disease, injury, or a faulty blood supply. This is also how you get disorders like age-related macular degeneration, AMD, or Alzheimer's. Dr. Kenobi also explains that these toxic aldehydes that result from omega-6 fats first react with hydroxyl radicals or peroxide radicals, producing a lipid hydroperoxide. This lipid hydroperoxide then rapidly degenerates into toxic aldehydes, of which there are hundreds, which in turn leads to cytotoxicity, genotoxicity, mutagenicity, carcinogenicity, obesogenic, atherogenic, thrombogenic, and teratogenic. When your body starts to break down all this excess linoleic acid omega-6, it gets broken down into harmful subcomponents called advanced lipid oxidization end products, or ALEs, and also oxidized linoleic acid metabolites that can cause significant damage at the cellular level. For example, an ALE called 4-HNE, which is 4-hydroxynonenal, is a mutagen known to cause DNA damage. Studies have shown that there's a definite correlation between elevated levels of 4-hydroxynonenal and heart failure. Linoleic acid breaks down into 4-hydroxynonenal faster when the oil is heated. This is largely why cardiologists recommend avoiding fried foods. Advanced lipid oxidation end products and the oxidized linoleic acid metabolites also play a significant role in cancer and heart disease. This is reference from Dr. McCullough. Today, our dietary ratio of omega-6s to omega-3s is approaching 25 to 1. The proper ratio historically for health has always been 1 to 1. And so we have an excess of up to, on average, 24 omega-6s that we do not have the antioxidant capacity to deal with this excess oxidation. When you look at the cellular toxins from the oxidation from these excessive omega-6 acids, you have the oxidized linoleic acid metabolites. They're mutagenic, which means they induce mutations. They're carcinogenic, which is cancer formation. Obesogenic is the cause of obesity. 4-hydroxynonenal is atherogenic, forming the fatty plaques in our arteries. Now, this has been blamed on cholesterol, and cholesterol is not the problem. It is the oxidized omega-6 within the LDL particle that is the problem. 4-hydroxynonenal is also thrombogenic, is creating blood clots. It's also genotoxic. This is damage to your genetic information in your cells. Malondialdehyde is mutagenic, again, inducing mutations. Cytotoxic, this is toxic to our cells. Acrolein, genotoxic, is damage to genetic information. Mutagenic, again, mutations. Acrolein is also teratogenic. It causes defects in the fetus. Carboxyethylpyrrole, carcinogenic, is cancer formation. And atherogenic, again, forms the fatty plaque in arteries. These are also known as foam cells. This arterial plaque is what cholesterol has been blamed for causing, so they can sell so many statin drugs. However, cholesterol is not the cause of arterial plaque. It is the oxidation of excess omega-6 leiolenic fatty acids that break down into cellular poisons and toxins that cause these toxic reactions within our cells. We have fats that protect our cells. We also have fats that damage our cells when in excess. Our stable, non-oxidizing fats, historically, they've been a balanced ratio of 1 to 1 with very little expression of disease. The oxidative process is required for energy production. We have to have it. But the antioxidation process required to rebalance it and detoxify the oxidation. And this is a critical balance. So whatever oxidation you have, we need the antioxidation to correct and rebalance it. A major change in our fat consumption. Highly unstable oxidizing fats. The last 100 years, with a very unbalanced ratio, causing 93% of the U.S. population to have metabolic disorders with increasing disease. Our imbalanced ratios now are in excess of 20 to 25 to 1. We have excessive oxidization and very little antioxidization. And this is not genetic. This is due to our dietary choices. Section 5, Summary. The omega-6 to omega-3 balance. Both omega-6 and omega-3s are essential fatty acids, and they must be present in our diet. Our bodies do not make these essential fatty acids. They must come from our diet. Omega-6 and omega-3s must be in balance, ratio of 1 to remain healthy. Both omega-6 and omega-3 play very crucial roles in our metabolic structure and functions. However, when these ratios are not in balance, we have a lot of metabolic damage and dysfunction. Currently, the American diets are loaded with omega-6 fatty acid, linoleic acid. This causes a lot of oxidation and damage. The breakdown of linoleic acid leads to mutagenic, carcinogenic, atherogenic, thrombogenic, genotoxic, cytotoxic, teratogenic compounds in our bodies. These compounds lead to many degenerative disorders and diseases.